Okay, so let's continue on the last two subtopic of chapter 2, which is gene technology. So the last two subtopic, we are going to look at methods, okay, of how to amplify DNA, and also how to observe, okay, DNA bands. Okay, DNA bands. So you have extracted genetic material from a sample of tissues, and maybe the sample of the DNA that you have collected, yang macam kita tengok semalam kan, you can collect DNA sample from semen, from blood, from bones, okay. So the sample that you collected and isolated the DNA or the genetic material may be low in amount, okay. So you can increase the amount of DNA that you have obtained in order for you to be able to analyze the DNA much better, okay. So untuk confirmkan result awak tu betul tak analisis yang awak buat tu betul ke tak. So in order to make sure that your analysis is precise, so you need to increase the amount of DNA, for example. So to increase the amount of the DNA you have collected is through the process of PCR, okay, polymerase chain reaction. So which is the last part. So after you have increased your DNA sample, you will run the DNA through a machine. We call it as gel or method. We call it as gel electrophoresis. Okay, gel electrophoresis is a method to analyze your DNA by producing various pattern of DNA bands. Okay, so yang semalam ni yang kita tengok yang DNA fingerprinting, how to get the DNA fingerprinting, you run your sample through this process, gel electrophoresis. Okay, gel electrophoresis. So through gel electrophoresis, you have your DNA sample. Okay, you have various DNA sample from individual 1, individual 2, individual 3. You have your sample of DNA, and then you are going to run the DNA sample through this method, gel electrophoresis. So your DNA sample will consist of various size or various mass, okay, that maybe you have cut using certain restriction enzyme, or certain that you have marked using certain markers, for example, okay. So through gel electrophoresis, you will separate the DNA into size, okay, separate DNA fragment by size or mass, okay. So the procedure or the method requires you to prepare gel. You have to prepare gel, okay. Prepare a gel that is made up of agarose or polyacrylamide, okay. So gel ni, kalau let's say lah masa awak final year degree ataupun master nanti awak buat ke whatever, PhD ke, yang awak terlibat dalam bidang genetik ke whatever, untuk awak analyze ataupun run gel electrophoresis ni, awak kena prepare gel agarose ni, kan. Dulu saya prepare gel agarose ni. Agarose ni dia adalah macam serbuk putih. Serbuk putih yang awak bancuh, guna ada buffer lah semua. And then awak bancuh dia, mixkan dengan buffer, and then put into a microwave. And then dia dah cair, dia dah jadi macam larutan jelly yang awak prepare, selalu prepare buat gel ke kan, buat jelly lah kan. So dia lebih kurang macam tu. So nanti akan ada satu acuan, kan, akan ada satu acuan bentuk persegi, nipis je, dia macam, tengok size sampel awak kan. So maybe gel tu half A4 size paper ke kan, half A4 size paper, dia punya acuan tu awak tuangkan gel tadi, larutan gel tadi, tuangkan dalam dia punya acuan dia tu. Okay, bekas dia persegi nipis je macam tu kan. Tuangkan, and then awak akan letak ada satu alat ni kita namakan dia comb. Okay, comb. C-O-M-B. Okay, awak akan tuang gel tu dalam bekas dia, and then awak akan letak satu plastik ni, dia nama dia comb. Okay, so kenapa nak letak comb ni? Supaya bila awak letak comb, dia tak appear lagi dalam screen kita. Okay, sekejap. Lambat betul dia appear. Nanti gel yang awak dah prepare tu, the gel that you have prepared will have well. They have wells. Lubang lah. Okay, they have wells. Alright. So, yang ini, okay, gel yang awak dah prepare ni, dia akan ada well, yang mana nanti awak punya DNA sample yang maybe awak dah run PCR, kan, PCR, you insert your sample A, sample B, sample C. Dekat hujung ni maybe adalah yang well yang awak prepare untuk standard. Okay, untuk standard. Selalunya kita panggil sebagai DNA ladder. Okay, so DNA ladder ni dia mengandungi DNA of known size. Okay, DNA of known size. So bila awak run, nanti dia akan produce what we call it as DNA bands. Ini yang akan jadi DNA fingerprinting nanti. Pattern dia berbeza. Cuma DNA ladder ni you know the size. Okay, you know the size. So maybe sample A dia akan bagi bacaan DNA dia kat sini satu, kat sini satu, kat sini satu. B, sini dengan sini. C, sini, sini, sini. Macam tu kan. So this the DNA band for sample A, for sample B, for sample C, you have to compare with the DNA ladder so that you know the size of the DNA. Ini dia bagi tahu size sahaja. Okay. Through this process, through gel electrophoresis, you cannot determine the DNA sequence. Awak tak tahu sequence dia apa, kan? So nak tahu sequence dia apa, you cut the gel that contain your DNA, you send to a company, that can analyze your DNA sample, that can give you the DNA sequence. Okay, so awak sendiri, awak tak boleh nak tahu DNA sequence tu. You have to send to a company that you pay that can analyze the DNA sample to determine the DNA sequence. Okay, cuma gel electrophoresis you run to get the DNA fingerprinting, the DNA pattern tu. Okay, so that you know. Okay, so yang atas ni size dia berapa, yang bawah size DNA dia berapa, yang tengah size DNA dia berapa, by comparing to the DNA ladder. Okay, so the gel that you prepare, before you insert your DNA sample, you must submerge the gel into a buffer, okay, that can carry current. So gel yang awak dah prepare tadi akan ada satu tank. Okay, akan ada satu tank yang mana awak akan letak your gel with the well. Okay, and then you pour the buffer inside the tank. Buffer tu akan tenggelamkan sekali gel tu. Okay, the buffer will submerge the gel also. Okay, so after you have placed the gel inside the tank and you have pipetted the DNA sample into each of the well, then you are going to connect the tank to electric source, okay, to an electrical source, electrical current. Okay, says here for the third point, when an electrical current is applied, the negatively charged DNA due to the phosphate group migrate towards the positive pole. So maksudnya bahagian yang ada DNA sample yang ada well tu tadi, awak connect dengan source yang bercharge negatif. Charge negatif dalam istilah sains apa? Dalam chemistry awak belajar, kalau negatively charged chemistry ke fizik ke kan, current yang negatif kita panggil apa?
Elektron.
Bukan elektron, yang untuk electric current, untuk run electric current tu. Itu ion. Cation, anion, cathode kan?
Anode.
Negatif cathode ke anode?
Anode.
Kejap, kita tengok Sekejap, nanti kita tengok istilah dia. The electric current where you have the DNA sample, awak akan connect dengan sumber negatif. The opposite and you letakkan dia dengan positif. Positive charge. Okay, positive pole. Okay, positive pole. Okay, so kalau kita tengok gambar rajah sekejap Oh, okay, betul lah. Anode, sorry. Alright. So, negative anode. Okay, so you run the current. Kenapa kat sini awak nak kena letak dia negatif pole dekat dengan DNA tu? Sebab DNA sifat dia negatif, kan? You know that the structure of a nucleotide, okay, dia ada phosphate, sugar, base. Okay, phosphate, sugar, base. The phosphate is negatively charged. So that's why, kenapa kita panggil DNA as DNA? Deoxyribonucleic acid. Okay, acid. Due to the presence of phosphate that is negatively charged, because in the aqueous solution, it is ionized. Okay. So, DNA bersifat negatif. RNA pun dia bersifat negatif lah. Okay, so bila dia bersifat negatif, when you run the current, you want the DNA, your genetic material to run towards positive. Dia akan pergi kepada positif. Daripada negatif, attracted to positive. Migrates to towards the pole, the positive pole. Okay, so you are going to apply electric current lah. So, the higher the voltage, the more quickly the gel runs. So selalunya you run the gel electrophoresis for about 1 hour to 1 hour and a half. Bergantung kepada size gel awak, bergantung kepada size sample awak. Okay. So kalau size gel besar, lamalah dia nak kena migrate. Lamalah DNA tu nak kena migrate daripada negatif ke positif. So you have to determine the right timing. So kalau awak run terlebih, nanti DNA tu akan migrate masuk ke dalam buffer. So sia-sia kerja awak kat situ kan. So, kalau awak terlebih masa or run the gel much more quicker due to the higher voltage, your DNA might end up in the buffer. Okay, so you have to find the right timing. Okay. So but if the voltage is too high, it can cause the gel to melt and also cause the formation of the band not so clear. You produce a smearing. Okay, you produce a smearing. Dia akan jadi macam smear macam tu. Tak berapa nak jelas macam tu. Okay, bila awak observe, right? And then larger fragment ni awak kena ingat. Okay, so the larger DNA fragment will move slower. The smaller DNA fragment will move faster. Sebab apa? DNA yang awak letak dalam well ni, through the electric current, it will migrate. Okay, dia akan start move towards the positive. So, yang akan bergerak dengan lebih cepat, obviously, the smaller fragment. So, the further the DNA sample from the well, the smaller the size. Smaller the size of the fragment. The nearer the DNA band from the well, the size will be bigger. Tu awak kena ingat lah. Okay, so, sebab apa? Bila DNA fragment tu besar, dia susah nak lalu dalam gel ni, so pergerakan dia lambat. Okay, the smaller molecule can travel faster, so it will be further away from the well. Okay, so after you have run the gel electrophoresis, you take out the gel from the tank. You take out the gel from the tank, and then you observe under UV light. Okay, dia ada mesin dia macam ada satu mesin yang awak akan masukkan dia. Mesin tu akan connected to a monitor. Okay, to a monitor. That machine will observe the DNA band that you produce using UV light. Okay, so sebelum awak boleh masuk ke dalam mesin yang observe under UV light, you have to dye. Okay, you have to dye your gel using dye lah kan, using dye, so that the dye can dye the DNA fragment. You cannot observe the DNA pattern, the DNA band using your naked eyes. Tak boleh. Lepas awak run gel electrophoresis, you cannot observe the DNA pattern. Awak akan nampak dia putih macam tu je lah, kan? So in order for you to observe this DNA pattern, you have to observe under UV light. Before that, you have to dye using a dye. Okay, to dye the DNA. Selalunya dulu kalau saya guna satu solution ni, we call it as ET-PR, ethidium bromide. So ethidium bromide solution dia sangat it is carcinogenic. So when you are handling this sort of work, you have to be very careful lah, supaya tak kena awak punya skin. Okay, so this work is very sangat rumit lah kan, sangat fragile. You are dealing with sample that you cannot see, kan. So you have to dye using ethidium bromide that is carcinogen. So you have to handle very carefully before you observe under the UV light. Okay, so that that you can observe through the monitor. Kat sini ada monitor. Okay, so the next slide. Okay, the DNA is visualized using a fluorescent dye that you can observe under using UV light. Okay. So here you have 3 reactions. Okay, reaction 1, reaction 2, reaction 3. Sampel lah kan, sampel DNA 1, sampel DNA 2, sampel DNA 3. The first sample of DNA that is in the test tube. Okay, awak letak dalam test tube sampel ni, and then you insert also restriction enzyme. Okay, restriction enzyme. So the restriction enzyme that you have mixed with the sample 1, so the restriction enzyme will cut the DNA into 2 fragments. One of short fragment and the other of long fragment, long size, bigger size. Okay, for sample 2, reaction 2, you also do the same. You put it in a test tube, you mix with the restriction enzyme. So the restriction enzyme will cut the DNA into 2 size of fragment. Okay, so the medium size fragment, and the other one is also the medium size. But if you observe carefully for the second reaction, you will notice that although dia tulis kat situ medium segment, the first segment here is much shorter compared to the second segment, kalau perasan. Okay, fragment yang pertama dengan fragment yang kedua. Fragment kedua lagi panjang sikit berbanding fragment 1. Okay, so for sample 3, you also do the same. You put the DNA sample in a test tube, you mix with a restriction enzyme. It produces 2 type of DNA fragment, short and long. Okay, so the DNA sample you insert into the respective well lah kan. So reaction 1, reaction 2, reaction 3. Okay, so you connect with the source of electric, so anode negatively charged, cathode positively charged, and then you run. So the DNA will move from negative to positive. Okay, the DNA will move from negative to positive. So the shorter fragment will travel further from the well. Ingat the shorter fragment will travel further away from the well. The larger fragment is nearer to the well. Okay, so after that, okay, you take out the gel, you dye using fluorescence dye, and then you observe under UV light. So this is under UV light lah, kan? So jap, wait until the next slide appear. Okay, So after you have run for gel electrophoresis, ini yang kita namakan dia sebagai gel electrophoresis. So gel electrophoresis, you place your gel with your sample and then you run the electric current. Okay, so it causes the DNA to travel according to the size along the gel. Alright. So after you run for gel electrophoresis, you observe under UV light. Okay, under UV light. Alright, so under the UV light, you will get this sort of pattern. Dia tak besar pun, awak tengok gel dia tak besar pun. Thickness dia dalam 5 cm, 5 mm, 5 mm macam tu kan. Tak tebal pun. Dia bergantung kepada awak punya size lah. Alright. So this is what we call it as the well. So well ni terhasil daripada comb yang awak acukan tadi masa nak buat gel. Okay, so dia akan ada ruang kat sini yang mana you will pipette your DNA sample during gel electrophoresis. So after gel electrophoresis, you will observe under UV light and you will get this DNA pattern, DNA band. Okay, so awak tengok the further away from the well, awak tengok dia punya fluorescence, the image of the band will be lighter, maybe due to the small size. Okay, the nearer DNA sample will be macam brighter, maybe of the larger mass. Okay, larger size. So kat sini you can place, kat sini sepatutnya ada DNA ladder lah, DNA ladder. Okay, so that you can know the size. Selalunya dalam several thousand base pair lah, base pair macam tu. Kat sini maybe 100 base pair, kat sini maybe 1000 base pair, 500 base pair, ini maybe 50,000 base pair. Okay, maybe. Okay, so by comparing the DNA ladder with your DNA sample, you know the size of the band. So the further away from the well, the smaller the size. The further away, so yang ni of intermediate size, yang ni of shorter size. Okay, the top one. Okay, so if you look at your DNA, your sample for this slide. Okay, sample 1. Awak tengok kat awak punya diagram lah kat situ kan, sebab saya punya dia transfer lambat sikit. Saya punya yang saya sketch, dia kata keluar lagi. Geram saya. Okay, so back to the diagram here. Okay, so okay, so this is reaction 1 tadi, reaction 2, reaction 3. Tadi dia kata apa? Reaction 1, satu dia akan hasilkan a very short DNA fragment. So this is the very short fragment and this is the long fragment produced. Reaction 2 tadi kita tengok dia akan produce 2 medium size fragment. Tapi saya suruh notice tadi satu lagi panjang daripada yang satu lagi. Satu pendek, satu panjang. Walaupun dua-dua of medium size. So dia akan produce satu strand yang atas, satu strand yang bawah. Yang bawah ni yang the shorter DNA fragment, yang atas ni is the longer DNA fragment walaupun dua-dua tu of medium size. Okay, for the reaction 3 yang hujung sekali awak tengok, so dia akan produce short fragment and the top one you observe is the longer fragment. Okay, yang reaction 3 tu yang belah kanan kalau awak observe. Okay, yang paling belah kanan tu. Okay, so you compare the DNA band kan dengan yang you observe cut using the enzyme kan, yang this slide. Okay, the previous slide. Okay, this slide here. So you just compare. Alright. Okay, so this is the video to explain on how gel electrophoresis works.
Enzyme 2 cleaves the DNA at a different recognition sequence. Adding both enzymes yields fragments. Both enzymes yield electrophoresis is one of the most useful means purifying DNA fragments for further analysis. In this technique, a jello-like slab DNA frag further analysis. In this cells, placed in a buffer solution and hooked up to positive and negative electrodes. The DNA solutions to blue dye are then pipetted into the wells. A well is also reserved for the placement of if no eye is turned on. The blue tracking dye is negatively charged and migrates toward the positive electrode, as does the DNA. The DNA contain negatively charged phosphates, which are attracted to the positive electrode. The smallest fragments move the fastest, tangled less at the base of the gel. When the blue dye reaches the bottom of the gel, the power is turned off. A fluorescent dye is then used to stain the DNA fragments. A DNA fragment in the size Stichter's lane is the smallest, in this case about 500 base pairs, followed by DNA fragment D. The largest fragment is 23,000 base pairs long. Each band of DNA may contain millions of fragments of the same size. In many cases, a researcher may want to determine which DNA fragment contains a DNA sequence of interest. To do this, the researcher prepares the DNA in the gel to make a copy, known as a blot. First, the gel is soaked in a basic solution so that the double-stranded DNA denatures into single strands. The gel is then transferred to a salt solution and a nylon filter is placed on top of the gel. Absorbent towels are placed on top of the filter. The salt solution draws the DNA through the gel toward the nylon filter, where the DNA adheres. The filter is treated so that the DNA adheres to it permanently, and then the filter is placed in a solution with a radioactive probe. The probe consists of single-stranded DNA that is complementary and will hybridize to the band of interest. The filter is washed to remove any unhybridized probe. A piece of plays the filter. The radioactive probe exposes the filtration. Knowing which band contain the sequence of interest, an identical ag gel and a band interest can be cut out of the gel. The DNA in this band can then be manipulated for further analysis.
Okay, so now we are going to look to the next part, the last subtopic of this chapter, which is PCR. Okay, polymerase chain reaction. Okay, so for polymerase chain reaction, PCR, it is a method to amplify and make many copies of specific DNA segments in vitro or replication of DNA in test tubes. Okay. Right, so that is the definition of PCR, polymerase chain reaction. It allows DNA replication to occur in test tube to make many copies. You have a small sample of your DNA. In order for you to make your analysis more precise, you have to amplify your DNA sample. Okay. Okay, so tools that are required in PCR, you will need your DNA sample, okay, PCR primers, as what we have learned during DNA replication. In order to synthesize the new DNA molecule, it must have primers. Dalam DNA replication hari itu, the primer was produced by the enzyme RNA Primase, kan? That is in your cell, but this one is using a machine. We are using a machine. So you just put in your reaction, okay? You just mix in the reaction the prepared primers, primer yang dah sedia ada, okay? So we call it as PCR primer, and then tag polymerase. Tag polymerase macam fungsi DNA polymerase tree lah dalam DNA replication. It adds the new nucleotide according to the DNA template. But in this reaction, in the PCR reaction, the enzyme that has similar function to DNA polymerase tree is tag polymerase. Okay, you add into the reaction the enzyme tag polymerase. And then the free nucleotides. The nucleotides we call it as DD and TPs. And short for apa? The many bases yang we have lah. Adenine, thymine, guanine, cytosine. Okay, the NTPs. We have DD, ATPs. Okay, DD, GTPs. Okay, so based on the bergantung kepada base tu lah kan. So secara short dia we call it as DD and TPs, free nucleotides. And then you need the machine. So the machine we call it as the thermocycler or PCR or DNA amplifier machine. Okay, so this is what the machine looks like. Dia tak besar pun, macam lebih kurang besar printer macam tu kan. So you have the button here for you to do your setting, nak run berapa lama, nak run berapa cycle, macam tu. Okay, berapa suhu yang the reaction requires, so you can set in this machine. Okay, suhu, cycle, berapa lama, macam tu. Okay, so the first tools that you need is the RNA primer here. Okay, so RNA primers are short single-stranded DNA sequence of around 20 nucleotide base pair. Okay, so you need two primers. Two primers are used in PCR, one for each template. So you have your DNA sample, and then the DNA sample will be separated to become template. Okay, so in order to synthesize the complementary strand, you add already in the test tube reaction the primers. So the primer will find the specific sequence to match on the template, okay, to initiate the process of elongating the complementary strand. Okay, so you have primer 2, primer 1 for each template. Okay, so the enzyme tag polymerase. Okay, so the enzyme tag polymerase, PCR, requires a DNA polymerase to synthesize the new DNA strand. So tag polymerase was isolated from the bacteria Thermus aquaticus, which is the prokaryotes live in hot spring. So that's why we make advantage of extracting the prokaryote enzyme to use in these procedures, okay, because it can stand high temperature, which is in the process of PCR, you will need to increase the temperature to 95 degrees. So what is the purpose of increasing the temperature to 95 degrees? So that the high temperature can break the hydrogen bonds between the DNA molecule. Okay, the function of increasing the temperature is to break the hydrogen bond between the two strands, so that the two strands can separate and becomes template. All right. So that's why you need an enzyme that can withstand the high temperature, which is tag polymerase. Okay, and then okay, for PCR, there are several cycles it must undergo. Okay, so it says here cycle 1. So produce 2 identical DNA molecule. Alright, for cycle 1, it involves 3 steps. Okay, 3 steps, 3 repeating steps yang sama: denaturation, annealing, and also extension. So the first step, okay, the first step is denaturation. What does the step involve? You increase the temperature at high temperature so that it is to break the hydrogen bond between the two DNA strands, so that the two DNA strands can become template. Okay, so original template of the target DNA exposed to high temperature of about 95 degrees Celsius. Okay, so to break the hydrogen bond between the two DNA strands so that they become template. Okay, so after denaturation is the second step, which is annealing. Okay, so annealing, you drop the temperature maybe to 60 plus degrees. Okay, 60 plus degrees. So the temperature you drop to this temperature, about 60 plus degrees, so that it allows primer to bind to the DNA template. Okay, DNA fragment is cooled to allow primers to bind to the strand. So this step we call it as annealing. After primer has bound to the DNA template, is the next step, which is extension, okay, or elongation. Okay, sama je, extension or elongation, where the free DNA nucleotides that are complementary with the original strand are added by Taq polymerase and rise the temperature to form a new DNA strand, produce two identical DNA molecules. You rise the temperature of about 70 plus degrees. Okay, does it give? So dia dah bagi dah kat sini. Okay. So these are the range of the temperature. For denaturation, the temperature is about 64 to 68 degrees. Okay, annealing is about 50 to 68 degrees. Elongation, you increase the temperature again to 72 degrees. So you must remember the temperature for each of the step. You must remember the sequence of the step. Denaturation first, annealing and extension, and you must know what happened in each of these step. Okay, so the next slide. Okay, so after 3 cycles, okay, so two molecules match the target sequence exactly. So this is the target sequence. Initially you have the target sequence. So after 3 cycles, kat sini you produce 2 fragments of molecules that are exactly the same as the initial molecule. Okay, but after 30 more cycles, you have 1 billion molecules that match the original cycle after 30 cycle. So that is the purpose of PCR, to increase or to amplify the amount of DNA from the original sample that you have obtained. Okay, so this is the video lagi. So I need to
copies of a specific. The polymerase chain reaction is a method for making many copies of a specific segment of DNA, starting with a very small amount. This technique can be used to identify specific microorganisms from a small amount of DNA and to identify persons involved in crimes from DNA on cigarettes or in a single hair follicle. The DNA to be amplified is mixed with deoxyribonucleotides, a thermal stable DNA polymerase. Is called Taq polymerase and DNA primers. The DNA primers hybridize to the ends of the gene to be amplified and provide a starting point for the Taq polymerase. The mixture is heated to break the hydrogen bonds in the DNA, forming single-stranded molecules. The mixture is then cooled sufficiently to allow the DNA primers to anneal to each end of the segment to be copied. Taq polymerase then synthesizes the complementary strand of DNA using the primer as the starting point. The temperature is raised again to separate the DNA strands and then lowered sufficiently to allow the primers to attach. Taq polymerase now synthesizes another set of new complementary strands. This process is repeated until enough DNA has been produced to be identified or used for further research. After 21 cycles, one molecule of DNA can be amplified to over 1 million copies. This amount of amplification can be achieved by running.
so that's the end of chapter 2.